Of all the interviews and podcasts that we've done, this is one that I am so unbelievably excited about, and one that I think that it's a harder discussion for a lot of people as we get into understanding water and deuterium and what that means. But I think maybe the first place to start is going to be like, why do the three of us even care about cancer? Why is cancer even on our radar, and why do we feel like this conversation is one of the most important ones we can have in regards to cancer? So, Petra, if you want to start, and then maybe go right into Gabor and why we respect him so much.
Oh, absolutely. So I think all of our lives have been touched by cancer. Literally, every single human being that I know has. And mine was touched actually very early. As you know, my dad died when I was 22. He was 63 from glioblastoma. So this is all very familiar to all of us, as of course your story is. And so when in 2018 I discovered deuterium uh, and its kinetic isotope effects. And then I understood there's ways that we can actually regulate deuterium and utilizing deuterium depleted water. And then I found, of course, Gabor's first book. I'm going to pull it out. That is very useful to first be introduced to this entire topic, and from there on we just built. And I actually started testing my clients as I shifted my entire practice towards utilizing deuterium regulation. I started using the testing, the prevent of water, and all of it, which at that time was available in the US. And we did a lot of patient testing that you processed for us. So that data, I all have it saved. It's been incredible. And your continued publications of showing the efficacy of using deuterium depleted water as a complementary tool for cancer patients has just shown amazing effects. So that's why we're here today.
My reason is that when I was kid, 12, 13 years old, our second neighbor had a head and neck cancer. And that was the first time as a kid I met with cancer. And it was very, very bad to see how the cancer progressed, and finally he died. And then I heard my parents and the others that they are talking about cancer. And then I believe that this is a serious problem. And that was more than 50 years ago, and it is still a serious problem. So it hasn't been done. So that cancer challenges. So that was the reason I started to think about cancer. And the next step was when the Albert Szent-Györgyi, who is one of the Hungarian Nobel laureate, at the 70s, he visited Hungary, and in an interview, he talked about that. He was a very, very clever. We talk about molecular biology, but it's impossible that this huge protein would be responsible for this very quick, very precise, and very harmonized responses. And he suggested that should be some submolecular level where the whole thing is organized. And he recommended that should be this small particle, the electron, which is a negatively charged small particles. So, and I kept it in my mind that submolecular, that was the key words. And Monday, that was 1976, May 21st, the idea came to my mind that maybe not the negatively charged electron, but the positively charged hydrogen should be that small particle. And then I kept it in my mind that the hydrogen should be. And that year, the next year I went to the university, and 4 years later, there came to my mind that there is a heavy hydrogen. It is called deuterium. That was in 1980. And at that time, I was sure that these two elements regulate the cell growth. That's it.
That's it. Off it went.
So what I'm so excited about bringing this conversation together is that that's a long time to be paying attention to deuterium, right? Like that's since the 70s. So Petra has a lot of the clinical experience. You've got an unbelievable amount of research and interviews and books you've written. So this is where we want to kind of bring together what is being seen with deuterium depletion and why that works, and what maybe we need to look at next. What's the next phase or next step that we need to understand? So, Petra, do you care to go into a little bit of what you see and how you utilize it, and then we'll get Gabor to kind of elaborate on that and what he's seen with his data and studies.
Yeah, absolutely. So I think it fundamentally restores the efficiencies of cellular metabolism and tissues, for that matter, and not just cancer, in all physiological processes and tissues. So whether that is thyroid function, whether that's for diabetes, it improves many different markers, and you've published papers on that as well, Gabor. And so uh, to me, it is a foundational tool, where we always should start first is addressing your deuterium levels. And I like to use food, of course, with that as well. If we reduce the foods that are high in deuterium from coming in, that's essential. Water, of course, can be extremely helpful, but not everybody can afford it. And then we also really have to pay attention to the environment and the light aspect and the sleep aspect and everything else that we know can contribute in regulating deuterium. And so it's been life changing for my clients, for sure. So yeah, it's been a wonderful.
Yeah. And the question is why the cancer problem has not been solved. And we know a lot of things about cancer. We know hundreds of genes involved in cancer growth, and we know thousands of mutations and the signal pathway, which has a key role. But the big problem is that the life is not organized on that level. And even targeting a tyrosine kinase and being very happy that there is a tyrosine kinase inhibitor, uh, we did not solve the problem because the system is so complex that even inhibiting one tyrosine kinase, there are 500 others, and the cell will manage it, that it doesn't count that there is a drug, so-called drug, and inhibit one path. So, for example, one of our study we published that we checked the expression of the genes, whether we can modify it, changing the deuterium concentration. And in that study, we tested over 200 cancer-related genes and over 500 kinase genes. And the point was when we kept the cells in deuterium-depleted media in normal and deuterium-enriched media, 300 ppm, we found that 97% of the genes responded to the deuterium-enriched media, which means if the DH level is higher in the cell, that will allow all the cells to trigger, to activate, and that way they can manage the cell growth. So when we are offering the deuterium depleted water, that means we can keep the DH level at a low level and will not allow the cells to simultaneously integrate and activate 500 genes, which is necessary to manage the cell cycle. And this is the reason that the deuterium depleted water is independently almost what type of cancer we talk about.
Mhm.
And we can get a very big efficacy what Petra has experienced, I guess, several times. And this is a big difference. So we are not targeting one signal system. We are simultaneously inhibit the whole system, applying and reducing the D level in the patients. And this is the reason that we say that that would be the breakthrough in cancer therapy, integrating deuterium depleted water, deuterium depletion into the existing therapy.
So, in from this farmer context, did you just say deuterium affects gene expression?
Sure. Yeah.
Okay,
Sure.
so if that's the case, I mean, that's obviously a big deal, right? Like the epigenetic factors, environment, deuterium itself, that's a big deal. So, Petra, what kind of a level do you see when a cancer patient comes into you and you test? What are those numbers looking like?
So they vary greatly, but they're all high. So high, I think maybe we should outline, the ideal kind of level. I believe for the human body is around 130 parts per million, and give or take, that can be between 125 and 135, if you will. But most of my patients, and initially when I was able to get both breath and saliva samples, that was really, really useful. But the highest measurement that we did actually was 161 parts per million in saliva, and that same person had 155.3 of breath sample. So I mean, that it's very significant, because we then also went and measured her water, because I wanted to know, where is this coming from? And so she actually had a level of deuterium above the natural abundance of the water that she was consuming, which ended up being 154. Still really high for the US, but it was definitely a conundrum. But generally, most people are in the upper 140s, 147, 148. Sometimes it's blatant in the 150s, but most of them are up there. So I think that dysfunction, the mitochondrial insufficiency, really truly starts above 140 parts per million in those samples that we've tested. I don't know, in your experience, Gabor, what you found with all of the people that you've tested over the years.
So what we have measured the patient's D level. So we have one way is to check it in the blood, but we had the much better system to condensate the exhaled air, the water from the exhaled, and that way we easily can follow the deconcentration of the patients. And making a kinetics with a couple of people, knowing what type of water they consumed and what is the D level in the body, that means we can we know what is the D level of the patients who consuming 105 ppm, 85 ppm, 65, or what. So this is the reason we mainly suggest what type of water you should drink, and based upon we know that what type of D level will result in the body. This is how we try to optimize the length of the DW consumption and the D level of the DW consumption. The starting point is in Hungary when we tested 30 people for diabetes. In the blood it was 146, 150 ppm, and that should be typical in Hungary because in that region this is the D concentration. Go ahead, Petra.
Yeah, I would love to actually expand on that for 1 second, take a sidestep, but we don't generally test blood levels at all. Can you expand why that is? And is there less variation? Is our body putting more deuterium in the blood? And what may be the function of deuterium in the blood? That I'm curious if you have any thoughts about that.
What do you mean? We do not?
So we don't measure generally the blood
Yeah.
levels in deuterium, right? So why is that? Is that not as reflective for us? Is that why we go for breath samples?
No, the reason it's invasive intervention when we are taking bloods. So we are not, I am not an MD and we are not an MDs. So we just giving advice to the patients who are receiving the conventional therapy, and we are going to help the patients and the oncologist with the DW. It is not my job to take the blood samples and all these things, but just collecting the exhaled condensate water, that's a non-invasive way. So that way we easily can collect and the samples and measure it. The other, we have facility to measure the D level of the water, but we do not have a mass spectrometer to measure the deconcentration of the blood. So this is the reason we are going that way. That the main focus is based upon the 33 years of experience with the cancer patients and following them depending on the cancer type, the staging, to give the advice which DW would be the best and how long should. This is the reason we are focusing on to get the best response.
Okay. Since you're mentioning the 32 years, 33 actually is for you now, right? So, but your latest preprint paper details findings that you took from patients all the way back to what is it, 1992?
92.
Yeah, to 2024. So that's a period of 32 years. And when I was looking at the data, I was wondering if you, because the numbers changed slightly from previous papers that you've published, where you stated that I think the median survival time increase was from 11.2 to 14.5 years, so 4 to 5 fold. So these numbers are a little bit different. And I was wondering if you could see from the data that over time, the medium survival time actually was less because of the changes in the environment that we've all experienced. Because clearly, somebody who lived in 1992 had a different environment than we have today, food wise, electric wise, all of that.
Yeah, so first of all, the paper was published, which is my peer-reviewed 5 days ago.
Yeah.
It is not a preprint. And so in that paper, we evaluated 2,649 cancer patients.
Yeah.
And our cumulative time was almost 10,000 years. And the aim was to figure out which are the basic parameters which are very important to maximize the efficacy of DW. And in that paper, we figured out 3 key parameters. The first, of course, how long the patients consume DW. And we say at least 90, 120 days is a minimum to see any positive effect of DW. The next point was to check what should be the survival time we need the patients should have when start the DW, and then we said it should be at least 4 months survival time. And the last was what can be the time frame between the diagnosis of the cancer and to start the DW consumption. And then we said that it should be less than 9 months. So if these parameters are optimized, in that case, we can increase the median survival time 11.6 years,
I see.
which is Hungary is 2.4 years for the whole cancer population.
Yep.
And answering for your question, so what I have seen earlier, we evaluated the median survival time of 180 lung cancer patients, and later we evaluated 300 lung cancer patients, and that increased. So as we are moving forward, the median survival time is increasing, and that depends on maybe because the patients come earlier to us, not in only the last stage, and maybe we can give more precise protocol when to stop, how long they should drink it. That can increase the median survival time, and maybe because the conventional therapy has some progress.
Sure.
Maybe I would say because the last 92 cancer drugs, which was approved by the FDA, increased 2.4 months, the median survival time.
Exactly. Yes.
2.4 months, not the
Yeah.
year.
Well, have you seen a correlation with deuterium levels and types of cancers? So what I'm saying is, does it seem like maybe pancreatic cancer is more associated with a high, and maybe, and I'm just throwing something out, a blood cancer is lowerly correlated? Is there any correlation that stands out?
No, this is a very good question, and this is what I have to investigate. So we should testing thousands of patients and healthy populations, cancer populations, patients with diabetes. And I think in that case, we can see some correlation that people living with a higher D level, what Petra said, over 150 ppm, they have a bigger chance to get cancer, they have a bigger chance to have diabetes. And you talk about epigenetics. So when we, Petra, you knows it well, that when we check the deconcentration of the different food, the carbohydrate results 150 ppm deconcentration, metabolic water, the fat 118 ppm. And I think the biggest problem in this whole society that 100 years ago, and my grandfather ate the bacon, and the people ate much more fat. So the calorie intake was covered not 70% by carbohydrate, but maybe 50 or only 30% by carbohydrate. And it means that the average D level of the society 100 years ago was much lower than it is now. And it means and this is what we say, that the optimal deconcentration for the mitochondria for the cell is 130, 35 ppm. And this is where the society is not there. We are at the 150 ppm, and this is over the optimal level. And we know that the life is how strictly regulate the sodium, potassium, magnesium, and nobody investigate the D level of the body. Nobody, but we must, because it has a key.
I'm curious to your perception around metabolic water, right? Because we have a lot of different terms that I think that we try to use. So you have Dr. Pollack talks about easy water. You have coherent domains. You have metabolic water. You have deuterium depleted. How do you view that terminology in your work?
I think that the metabolic water is when the hydrogen is coming from the carbon source, amino acids, or lipids. And when the mitochondria burn the food to carbon dioxide and metabolic water, in that case, the hydrogen is coming from these carbon bond, glucose, different amino acids, or the lipid. And it means earlier the plant synthesized the sugars, or the animals ate the carbohydrate, prepare lipids, and we ate the meat. And in that case, it has a lower D level. And finally, that D level will appear in the mitochondria when it produces the metabolic water. And 0.3, 0.4 liter water, which is synthesized by us every day. And depending on how much water you drink, that will give the average D level. So we do not recommend people drinking 3, 4, 5 liter waters, because it means they are drinking 150 ppm and producing 0.4 liter deuterium-depleted metabolic water. The average will be high. If somebody drink 1.5 liter, the average will be much lower, and that will modify, and that is determine, I guess, the whole metabolism and the chance to get cancer or not.
Petra, you got to follow up?
Yeah, I mean, I've heard different numbers of the amount of water that we produce, and it's likely much in excess of what you just suggested, 0.3 mLs or 0.4, right? But regardless, it is this metabolic water that's absolutely key for us, and I totally agree that deuterium loading with bulk water, whatever water that comes from, is inappropriate. We should really only drink water based on thirst, and it's a key aspect for sure.
Yeah.
We are currently doing a little experiment with basil plants. So there's like 8 little basil plants, and we have some with deuterium depleted water, some with distilled, some with spring water. And it's really interesting that the deuterium depleted water has a more compact structure. It's greener. It's really interesting. And whereas the more of your like tap water or even distilled, it's more spindly. It's a lighter color. It's a lighter green, almost a yellowish hue. And so there's obviously a massive difference in the plants with deuterium too. So do you have any insights on that?
In Hungary, at the university where I graduated, they run several experiments and checking how the germination, how fast. And they found that in deuterium-depleted water, at the beginning, it inhibited and slowed down the, the, the growth of the roots and the sprout. And later, after 1-2 weeks, they adjusted the system, and then after that, there wasn't any experiment, the differences. And the other paper we published, we used the allodea. This is a aquatic plant, so living in the plant. And the experiment was that from the allodea was removed from the normal water and put it into the deuterium-depleted water. And immediately the allodea started to transport the hydrogen out of the cell and became acidic outside. So the membrane transport is so sensitive for the DH ratio that immediately activated it. And after half an hour, they go back and they adjusted everything to the lower D level. So this is what we are doing with the patients. They are drinking DW, we are challenging all the cells, cancer cells and healthy cells, and hopefully the healthy cells can handle it and the cancers cannot. If the dosage is good and the D level is good, then we can select the cancer cells because they are not able to adjust the whole system to the lowering and changing D level.
So, since you're mentioning that, I would love for you to expand a little bit on this principle of cycling the different ppms over time, and how did you determine that kind of time interval and the 20 ppm drop, and then yeah, I would love to know, because that's a question we often get, and you did a great job writing some of these protocols in your latest book, but I'd just love for you to talk about how you figured out that that would be a useful way to approach drinking DDW.
Yeah, so when we started, we had only one type of water, and so it started in 1992, and since the next 8 years, there was a time when we had a very low D level, so we just tested. Of course, I didn't know anything about at the beginning, just somebody started to consume, and I started to collect all the information. So the basic concept is when somebody, okay, let's say make two groups, three, healthy people, cancer patients. The healthy people, we recommend to consume 105 ppm or 125 for 3-4 months every year, every second year, every third year, depending on the risk factor, just to reduce the incidence of cancer. Because we know that everybody has cancer cells, but this is balanced with the immune system, and it's fine. So when somebody consume DW for 3-4 months, that means will be a reduction in the D level, and hopefully we can select almost all of the small group of cancer cells. And that way we should reduce the incidence of cancer. For those who had been diagnosed with cancer, we can make two groups. One, they were treated and they are tumor free, and the other, they are receiving the therapy and they has a cancer.
Yeah.
So the best way would be to reduce the cancer mortality if we be targeting those who are in remission. So they were diagnosed, they were operated, they received the chemotherapy, radiotherapy, just hoping that they kill all the other cells, which was not removed by the surgery, and that way they will be in remission after 20 years. But everybody knows the cancer come back. So to reduce the chance of the relapse, we recommend for cancer patients being in remission to make a 2 months 105, 2 months 85, and then have a break. And those who are close to the complete regression, we recommend maybe they should drink longer, this 105/85, and the next break should be shorter. So 4 months cure, 4 months break, start again with 4 months, 105, 2 months, and then extend. The next break should be 6 months, and then start again and start again. That way we can prevent the relapse. We never knows whether we have cured all the cancers, but repeating it for several years, it is a very good chance. So our number shows, for example, we published breast cancer, 48 breast cancer patients being in remission. The cumulative follow-up time was over 200 years, and we lost only one patient. The other we published which represented over 260 cancer patients and representing all kinds of cancer, but they were in remission. And the cumulative follow-up time was over 1,200 years, and we lost 18 patients out of 260. And the point is that 8 of them died. They never came back. They were tumor-free. They consumed DW. Then I heard that the average time was 4 years later they died. Those who coming back and repeating the DW cure for several years, they still in remission.
Yeah.
So this is the group of people who are in remission.
So we just point out we can't call it cure in America, right? Because none of it cures. It's an adjunct, or
I know.
yeah. But that is what you call it in Hungary. But I'm still just want to understand why you actually dropped down the 20 ppm from 105 to 85. How did you assume that was the right? Why not 30 ppm? Why 20?
No, I don't know that. No,
Okay.
I don't believe that this is the last protocol what the people will forever use.
Okay.
So when at the first we had on a large scale, we could produce it was 105 ppm.
Yeah.
And at that time, everybody consumed 105 ppm.
Okay.
And then we came out with the 85 ppm. But what I can say that the 20 ppm drop is important, because I know that there was a couple of people who started to mix it, or they didn't follow the protocol, and I saw that it wasn't so effective. And the other, checking the kinetics, how the D level is changing in our body, we can say that after 2 months, we do not cause any changes in the D level, because there's the equilibrium and no change after that. But we need to challenge the cancer cells, and the way we can challenge it, we go a lower D level, and then there's again a big changes, and that hopefully select the cancer cells.
Yeah, so really that becomes extremely important when you are dealing with an active cancer, specifically, right? But for prevention, I would think there is a little bit more leeway in how you could go about it.
Yeah.
And certainly if you add other pieces like food and light and all of that. So yeah, that's interesting.
One thing.
The third group is a other story, that what type of cancer we talk about, what is the stage, what type of commercial therapy they receive. So what is important, always combine DDW with radiotherapy. That can significantly increase the efficacy of the therapy. So combining these two. For example, with glioblastoma, when it was combined, the radiotherapy with DW, the median survival time was 47 months instead of 12 months. So we get a 3-fold increase in median survival time.
That's amazing.
The DW can be important to give the patients before the operation, because that way the operation will be much easier, and the tumor will be removed much properly. And we also can see some good synergistic effect with targeted therapy, because finally they are stopping and inhibiting one signal pathway, and that can help the efficacy of DW too.
one of the aspects that we struggle with is cancer's ability to hide or deter the immune system, right? So with the deuterium being a likely contributor to that, do you think this is from the glycocalyx? Or are they using, is cancer using deuterium to strengthen itself? Why is lowering deuterium making it more susceptible to some of the chemo radiotherapies? Do you have any thoughts on deuterium and the hiding from the immune system that cancer does?
Yeah, so what we say that the signal for the cell to multiply when the membrane transport is activated and exporting the hydrogen, but this system will prefer to eliminate the hydrogen, so can increase the DH ratio in the cell. The cell can start at proceed from the G1 phase to the H phase when the DH level is pretty high, that activate all the enzymes which are necessary to duplicate the DNA and then G1, G2, and M phase. So this is without increasing the DH level to the threshold, the cell will not multiply. This is what we state. And this is what we said in the very first paper in 1993. A higher DH level is necessary to trigger the cell growth. So, and that can occur when a growth hormone binds to the membrane, and that will stimulate the sodium hydrogen antiport. And the cancer is cancer cells that because the damaged mitochondria. So in a healthy cells producing deuterium-depleted metabolic water, this is a break in the system. So even the membrane transport is going to increase the DH ratio if the mitochondria in a healthy cells working properly, producing DW metabolic water, so will not allow to the cell to lift up the DH ratio to the threshold which is necessary to initiate the cell growth. And this is the reason that the cancer cells, no break in the system. So the mitochondria is damaged, no TCA cycle, no metabolic water production with a low D concentration. So this is a basic mechanism what our science and what our research says.
I want to add to that because I think there are additional mechanisms. And the last year, I have been studying the glycocalyx, the endothelial surface layer complex. What I've learned is very interesting because you're just describing intracellular processes, but I think this body of research, and I've learned a lot from Barry Ninham and his colleagues that have published fascinating papers on this, that there is a endothelial surface layer glycocalyx complex around every cell, every tissue. It lines every vasculature, anything. And so the thickness and the function and the ability of anything of cellular trafficking, if you will, is all determined by this layer. And not only that, the bottom layer of it, it's the glycocalyx that's made up of these water tubules that are these nanotubules, and they're made from water. And so the amount of deuterium present in that determines the curvature, the conductivity, and the connectivity of this network. And it's a nano network that also functions as a neurological network. And so depleting deuterium with deuterium depleted water will actually thin that layer out and provide access to the cell. And so I think that's a whole other aspect that will be so much fun to explore.
Yeah, I agree with that. So all the physical parameters can influence what's going on in that. So one, for example, I always there is a big demand whether it's a genetic origin of cancer or metabolism. So, and I think that there is a very strong connection between the genetic background and the metabolic. Because, for example, when the EGFR is amplified, so it means there are 40 times more receptor in the membrane, which is the reason is a genetic problem. But if the receptor number is 40 times, 100 times higher than the average, in that case, the growth hormone more has a more strong effect to stimulating the sodium hydrogen antiport. And that way, the cells can very easily lift up the DH ratio to the threshold. So there is a genetic changes, and that can influence how the DH ratio can modify. And all the other parameters surrounding the cells, what you say, it's everything can influence it. And again, what you eat, what is the composite, how was it hydrolyzed, what is the source of the food, and what was the deconcentration of the water, which was the plant was watering. Everything depends on.
Right. Yeah. I mean, there's so much more to it, I think. And maybe, Logan, you want to introduce, some of what we've been talking about and what we've written about, but the photo bioelectric magnetic aspects really are the key to this entire story of cancer. And we think deuterium is an integral part of that, but ultimately, all of these additional biophysics processes are absolutely fundamental. It's not just about genetics and metabolic theories anymore. We can't say that, I think.
Finally, everything is running on a according to the quantum physics. So everything what we see is the final production of billions and billions chemical reaction where the rule is governed by the quantum physics, and that's it. So it is so far from the molecular aspect of the drug development, and very, very far. But
That
this is the
sub-molecular
basic
you
of the
talked
life.
about. Yeah.
Yeah.
It's fascinating. And so what we have found with there's so many brilliant people that are working on different parts of that elephant, so to speak. And when you start seeing a little bit of that crossover and this what I refer to as like a lattice work of mental models, it's really really cool. And when you see the deuterium story with uh the geographic distribution, right? Like higher towards the equator, lower towards the poles, what that happens. And so, like, one thing that we dove into and I think is really, really fascinating is the whole egg aspect. So, in the egg whites, we have higher levels of deuterium. I believe that that has some sort of a electromagnetic protectant, right? We've been talking about the negative aspects of deuterium, a positive aspect, and it protects the yolk from the environment. It does some other things. So, like the looking at say an ostrich versus a penguin, and then the concentration of deuterium within the egg whites. I think there's a lot of this is theoretical, right? Like I'm trying to process this, but it makes a lot of sense why we would need different deuterium levels in different parts depending on where we live and what time of the year, right? So I think that this is kind of where Petra was talking on the blood side of it, and the medical intervention makes perfect sense on why that's not been the way you've done it. But I almost think that there is a benefit of deuterium in the blood, whereas in the cellular matrix, it's a disaster that leads to cancer, right? So do you have any thoughts on that? Like maybe and this may be crazy, right? The positives of deuterium and how it's utilized in the body?
I would say that without deuterium we wouldn't be here. So I so so when we checked the effect of deuterium on bacteria, we could not see any effect in growth and anything. So I think when during the evolution the life was getting more complex, and more information was packaged into one single cell, into one single nucleus, and became more and more complex. There was a time when the molecular level they were not able to harmonize it. And when the bacteria moved into the eukaryotic cell, and that was the mitochondria, then will be the chance producing DW by the mitochondria and the membrane transport, and there then this submolecular level somehow, so the life used out that there is deuterium and hydrogen. I do not say never that the deuterium is dangerous and we should remove it. I say it is a very important part of the regulatory system. And the question is whether we can manage it and we are doing it on the right way. And our food and food industry and all these things, whether take care of it, they don't, because nobody take care of the D level. So, and one thing, 40 years ago, I made a dissertation, some kind of, we are thinking about the fission energy. So putting together the deuterium and using the to generate electricity. I don't know, but do I see in the right way? So we are going to use up the deuterium from the nature to produce electricity. And we are using up something which is necessary for the life. 150 years ago, we started to burn the oil and coal and all these things, and we increased the D2O level, CO2 level, and now our society is working on to remove and use up the deuterium just to produce enough energy. So the deuterium is essential to keep that very complex thing, what we call life.
That I love it. I love it. So one thing as I was trying to understand deuterium was I came across a deuterium lamp, right? This is a UV lamp. So there is some correlation there to where when deuterium is electromagnetically squeezed, right, it can produce a UV light. So when you tie into, Roland Van Wyck's work around biophotons, like you said, we wouldn't be here without deuterium, right? So like I think that there's so much of that maybe we're it's an exciting time to be alive, and I don't even know what we're about to find out. But like the idea of squeezing deuterium to produce UV light in relation to these biophotons just sounds really incredible to me. So, any thoughts on that?
No, I am not familiar with that, and I wouldn't like to say something which is far from the reality. No, but again, so when we receive the light, of course, it influences our body and our cells recognize it and producing vitamin D. So just because we receive the light, of course, it generates lots of biochemical changes, and of course, the deuterium can influence that process or vice versa.
Awesome. Petra, what you got?
So I have one more topic that I like to cover with you. A very frequent question I get from my clients who are, they tend to study and they tend to listen and they are informed. And so they will come to me and say, I heard Dr. Somlyai say on another podcast that you are never supposed to have any antioxidants when you're drinking deuterium-depleted water.
That's true.
So,
Yes.
so I want to go there because I on my website I've listed I think 18 different mechanisms now by which deuterium depleted water can actually benefit us, and I think only one of them is about causing this oxidative stress piece, but there's many others. But I want to make this case, and I'm wondering how you would think about that. That first of all, we should do what Cameron Borg has suggested, call antioxidants is way too much of a broad term, and it's not really correct. We should call them redox modifiers. And there's very big difference between what we have available to us. And there is amazing data on botanicals that we probably would consider antioxidants, if you will, whether that's resveratrol, whether that's turmeric, whether that's apigenin, and there's just so many of them, beautiful compounds, milk thistle and so forth. And there's great data that shows that it actually increases apoptosis when you are on chemotherapy, that it protects the healthy cells from the toxicity and so forth. So to me, it's my plea to you, because so many people come, no, I can't take anything. I think that's not it's too general of a term, and I think I totally agree with you on any type of synthetic supplements like glutathione, like there's so many, the
Vitamin
synthetic
C.
vitamin E's, all of it. Yes, I agree with you on that, but there is nuance here. And so what what do you have to say about that?
I understand. So what? So the only thing I did, I talked to patients from morning till night for several years,
Yeah.
and there was two very specific groups. One group which responded extremely well, and sometimes I found that they did not respond so well to deuterium depletion.
Mm-hmm.
So the question was why they do not got so good results, why we do not see good response, big reduction in the tumor size and all the thing. And finally, I identified three parameters. The first, those who taking the antioxidant vitamin C, A, E, selenium, it was clear that 2 months ago, a year ago, that was common in that patients, they took too much antioxidant.
Mm-hmm.
And the fact which confirmed me is published by Zubarev. So they published that deuterium depletion trigger radicals, and that radicals triggered apoptosis. So those who are taking antioxidants, the antioxidants will catch the radicals, and the radicals level do not reach the level which would trigger the apoptosis.
Right.
So this is the reason I didn't figure it out. I just followed the patients and tried to figure out what is the reason that do not respond so well. The other was when, for example, there was a lady who was in a late stage breast cancer bone metastasis. She started to dig DW, she was fine. Then she went out to the garden and dig the garden, and then he relapsed, and we lost it. The other guy had a lung cancer, he became very fine, and then they make very strong physical activity, and then he relapsed. So the other parameter, we do not recommend people to make exercises, to lack of oxygen and wetting, because that changed the metabolism of the cell, the lactic acid appear, and somehow that will help the cancer cell to survive the deuterium depletion. So that was the second one. The third one was the hot bath and the sauna. So I had the same type, that was very accidental, that the same type of tumor, Hodgkin lymphoma, one of them took a hot bath, the other a cold bath. The hot bath died, the cold bath, he responded quite well. When we increase the body temperature again, that will modify the metabolism in the whole body and in all the cell. And again, that somehow is help the cancer cells to manage uh, the challenge of the deuterium depletion. And yeah, that I wrote in the book the goat milk, because the goat milk has quite a different composition of the amino acids, and also think that the vegetarian diet is not good, because the vegetarian diet is good because, again, a lack of amino acids, and the cancer cells fear that something is not ready, so they are not in the sensitive phase of deuterium depletion. So this is the reason we say that the patient should eat animal proteins, eggs or meat or something having enough protein, just to mimic that everything is fine. So the cancer cell recognize, okay, I can duplicate the proteins, but will not be enough deuterium. So these are the consequences, what I say, of the long term of discussion with the cancer patients.
Right. Okay. That's
And
helpful.
on the other, that's very important. Vitamins are important, and vitamins need. And I do not say it should be zero because we cannot do that, but I don't know what is the minimum D vitamin which help the body, but not help the cancer cells to somehow tolerate the deuterium depletion.
Right. No, that
So
makes
and
sense.
it's a
Thank you.
very fragile system to
Yeah,
we need to
absolutely.
balance.
Yeah.
Yeah.
So what do you hope to figure out next? What's the next step for you?
I don't know whether you have seen, we just yesterday, there is a news release in the United States in the Yahoo News, Yahoo Finance, and Business Insider. So my dream is, so in that news release, we state the conclusion of the last paper that 75-80% of the cancer patients' life we can save it. So the cancer mortality we can reduce it by 75-80% if the deuterium depletion would be part of the therapy. And nobody can say that. No any cancer drug development say that because they talk about, okay, we increase the median survival time with 2-3 months. So that should be a breakthrough. If the world recognizes the potential of that, and then everybody would work on it to put deuterium depletion part of the oncotherapy. So I don't know what will be the consequence of this news release. I have seen lots of paper cited it. We are waiting for investors, waiting for pharma companies to move forward, to spread, to share the knowledge, to reach more cancer patients, and one day I hope the world will accept it that this is really new and we should move into that direction. And it's very important. We are not going to replace the existing therapy with deuterium depletion because that won't be the best for the people. We have to integrate deuterium depletion to the existing therapy. That is the best way to move forward.
Yeah. Thank you, because your work has been instrumental in my practice in the last
You are
7
welcome
years.
at any time. Just let
Yeah.
me know.
Thank you.
Thank y'all so much. I think that this was power packed with a lot of information. I think that deuterium depletion in the right protocol should be incorporated in every journey, right? And there's a lot more that we can go into. Only so much you can in a short amount of time. But y'all two are like the deuterium expert. So like I think that this if you're interested in deuterium, this was the podcast to at least dip your toes into. So again, thank y'all.
Okay.
Thank you.
Thank you for having me. Take care.